Hi viewers, in this class we are going to discuss the MHC Major Histocompatibility Complex. Under this we are going to discuss introduction, general organization and inheritance. MHC encodes three classes of molecules, allelic forms of MHC which are usually known as haplotypes, Class I MHC molecules, Class II MHC molecules, Class I MHC peptide interaction, Class II MHC peptide interaction, and the Class III MHC molecules and their respective examples. See the introductory part of the MHC. T cell receptors only recognize antigen that has been processed and presented in the context of molecules encoded by the major histocompatibility complex or MHC. The MHC was first studied and so named as a genetic complex that influences the ability of an organism to accept or reject transplanted tissue from another member of the same species. Here, histo refers to the tissue and compatibility refers to living together harmoniously. An MHC molecule always recognizes only T lymphocytes. The two types of MHC are worked in immunity. T helper cell recognized by MHC class II molecules and cytotoxic T cells are recognized by class I MHC molecules. Usually, major histocompatibility complex is set of surface proteins located on the cell membrane of nucleated cells. It plays more important work to identify the antigen between self and non-self body, intracellular recognition, and responsible for antigen presentation. Here we are visualizing the two legendary personalities, Peter C. Doherty and Ralph Martin Zinckernagel. Pioneering studies by Ralph Martin Zinckernagel and Peter C. Doherty and others made it clear that molecules encoded by the MHC play a seminal role in determination of adaptive immune responses, and the particular set of MHC molecules expressed by an individual influences the repertoire of antigens to which that individual's T helper cells and T cytotoxic cells can respond. Rolf Martin Zinckernagel received Nobel Prize in the year 1996, shared with Peter C. Doherty for the discovery of how the immune system recognizes virus-infected cells. Viruses infect host cells and reproduce inside them. Killer T cells destroys those infected cells so that the viruses can't reproduce. Zinckernagel and Doherty discovered that in order for killer T cells to recognize infected cells, they had to recognize two molecules on the surface of the cell, not only the virus antigen but also a molecule of the major histocompatibility complex, that is MHC. This recognition was done by a T cell receptor on the surface of the T cell. The MHC was previously identified as being responsible for the rejection of incompatible tissue during transplantation. They discovered that the MHC was responsible for the body fighting meningitis viruses too. And coming to the general organization and inheritance of MHC. Every mammalian species studied to date possesses the tightly linked cluster of genes that constitute the MHC, whose products play roles in intercellular recognition and in discrimination between self and non-self. Here, if you observe the history, Peter Gorer found that four group of MHC molecules used the blood sample of mice to identify the blood group antigens which designated by Roman number 1 to Roman number 4 group of MHC. George Snell and Jean Dosset and Berry received Nobel Prize in 1980 for their contribution to the discovery of the MHC molecule. See, it was the Peter Alfred Gorer and Burroughs Binasarov, B. Binasarov, Jean Dosset and George D. Snell discovered the genes that regulate immune responses. Immune response gene, now known as the major histocompatibility antigens, they were awarded Nobel Prize in 1980. How to define the MHC molecules? Major histocompatibility complex is a membrane-attached protein which work on recognition of antigen between self and non-self body and antigen presentation. Whereas here there is a MHC complex. Molecules are the surface proteins. Whereas MHC, that is major histocompatibility complex, is a large genomic region or gene family found in most vertebrates. It is the most gene-dense region of the mammalian genome and plays an important role in the immune system, autoimmunity, and reproductive success. The MHC participates in the development of both humoral and cell-mediated immunity responses. The cluster of genes responsible for the rejection of foreign tissue is a result of an immune response to cell surface molecules. They are usually called as the MHC complex. In mid-1930s, Peter Gorer was using inbred strains of mice to identify blood group antigens, identified four groups of genes designated as Roman number 1 to Roman number 4 that encode the blood cell antigens. Work carried out in the 1940s and 1950s by Peter Gorer and George Snell established that antigens encoded by the genes in the group designated 2, Roman number 2, took part in the rejection of transplanted tumors and other tissues. George Snell called these genes histocompatibility genes. Their current designation as histocompatibility 2, H2 genes was in reference to Gorer's group 2 blood group antigens. Although Gorer died before his contributions was recognized fully, George Snell was awarded the Nobel Prize in 1980s for his work. And MHC encodes three major classes of molecules. The major histocompatibility complex is a collection of genes arrayed with a long continuous stretch of DNA on chromosome number 6 in humans and chromosome number 17 in mice. The MHC is referred to as the HLA complex in humans and the H2 complex in man. HLA complex in humans and H2 in mice. Although the arrangement of genes is somewhat different in the two species, in both cases the MHC genes are organized into regions encoding three classes of molecules. This is the HLA, human leukocyte antigen, and here this is the chromosome number 6, and the HLA antigen region which encodes class 1, class 2, class 3 MHC molecules. This is the HLA region, and this is the chromosome number 6. See here, chromosome number 6, genes for class 1 region, class 2 and class 3. So these genes undergo transcription and translation forms the proteins which are present over the surface of the cells. They are nothing but the MHC molecules. Here we have to understand that MHC, when we talk about the major histocompatibility complex, it is a gene complex upon the chromosome number 6 of the human beings, which is nothing but the HLA gene complex. And when we are talking about the MHC molecules, they are the membrane bound surface proteins. We have to discriminate the difference in between MHC, that is complex, and the molecules. Here we are discriminating the difference in between human HLA and mouse H2 complex. The type of genes and their loci, both of them are different. And all these genes which are present in the MHC locus are tightly linked and usually inherited together. And coming to the class 1 MHC genes. Class 1 MHC genes, they encode glycoprotein expressed on the surface of the nearly all nucleated cells. The major function of class 1 gene products is presentation of peptide antigens to cytotoxic T cells or CD8 cells. See, this is the organization of the class 1 MHC genes. Here you can observe, this is the DNA molecule and it is underwent transcription from the mRNA, and here alpha 1 gene for alpha 1 region, alpha 2 and alpha 3, and here TM means transmembrane region, and this is the cytoplasmic region. So the amino acids which are present in each and every region, like alpha 1 encoded by alpha 1 genes. So alpha 1 genes undergo transcription and forms the amino acids of the alpha 1 region of the MHC class 1 molecules. Like that we have to understand. So these are the genes responsible for the synthesis of class 1 MHC molecules. Then coming to the class 2 MHC genes. They encode glycoproteins expressed primarily on antigen presenting cells, that is ABC, such as macrophages, dendritic cells and the B lymphocytes. They present antigenic peptide to T helper cells, otherwise CD4 cells. Here you can observe the organization of the class 2 MHC gene. Here this is the DNA and this is the mRNA. DNA and mRNA. Here the beta 1 synthesizes beta 1 gene complex synthesizes beta 1 chain, and beta 2 transmembrane and as well as the cytoplasmic. Here also alpha 1 alpha 2 transmembrane region and the cytoplasmic tail. Like this, there are different regions, gene complexes in the DNA that underwent transcription from the mRNA, and they are translated into the specific regions of the MHC class 2 molecules. And class 3 MHC genes, these genes encode, in addition to other products, various secreted proteins that have immune functions, including the complement components C4, C2, they are the complement proteins, and factor B, and several inflammatory cytokines including TNF. TNF is the tumor necrosis factor. And coming to the class 1 MHC molecules, they are encoded by the K and D regions of in the mice, whereas A, B and loci in humans were the first discovered, and they are expressed in the widest range of the cell types, so that they are known as the classical class 1 molecules. classical class 1 molecules. Additional genes within the H2 or HLA complexes are encode class 1 molecules. They are known as the non-classical class 1 genes. Non-classical class 1 genes and the classical class 1 genes. And here functions are not known for all these gene products. Some may have highly specialized roles in the process of immunity. If you see the best example for this, the expression of class 1 HLA G molecules on cytotrophoblasts at the fetal maternal interface has been implicated in protection of the fetus from being recognized as foreign. This may occur when paternal antigens begin to appear and from being rejected by maternal cytotoxic T cell. This is the best example. Then here you can observe, this is the ACB or ABC genes codes the class 1 MHC molecule in the human beings, whereas DL they encode for the class 1 molecules in the mice. So the gene complex of human beings is HLA and gene complex for the mice or mouse is the H2. Class 1 MHC locus. So D, K and the L. And coming to the class 2 MHC genes encoded by the IA and IE regions in mice and DP, DQ, DR regions in the human beings. DP, DQ, DR in the human beings. See here, class 2, DP, DQ, DR. And here class 2, IA, IE. And here these are the genes for class 3 locus and these are for the class 3 locus in the human beings. And coming to the in general MHC molecules, class 1 and class 2 MHC molecules have common structural features and both have roles in antigen processing and the presentation. Whereas the class 3 MHC region is flanked by class 1 and class 2 regions encodes molecules that are critical to immune function but have little in common with class 1 and class 2 molecules. And come to the class 3 MHC genes. Class 3 products include the complement components, that is C4, C2 and factor B and several inflammatory cytokines including the TNF, tumor necrosis factor. And coming to the next one, allelic forms of MHC genes are inherited in linked groups called the haplotypes. They are called as the haplotypes. Here, the loci constituting the MHC are highly polymorphic. That is, many alternative forms of the gene or alleles exist at each locus among the population. Most individuals inherit the alleles encoded by these closely linked loci as two sets, one from each parent. Each set of alleles is referred to as the haplotype. An individual inherits one haplotype from the mother and one haplotype from the father. In outbred populations, the offspring are generally heterozygous at many loci and will express both maternal and paternal MHC alleles. The alleles are codominantly expressed, that is, both maternal and paternal genes products that are expressed in the same cell. And if mice are inbred, that is, if mice are inbred, have identical alleles at all loci, Each H2 locus will be homozygous because the maternal and paternal haplotypes are identical and all offsprings therefore express identical haplotypes. Certain inbred mouse strains have been designated as prototype strains, and the MHC haplotypes expressed by these strains is designed by an arbitrary italic superscript, example the H2A and the H2B. These are the prototype strains. In an outbred human population, each individual is generally heterozygous. Individual is generally heterozygous at each locus. The human HLA complex is highly polymorphic and multiple alleles of each class 1 and class 2 gene exist. The human HLA loci are closely linked and usually inherited as haplotypes. When the father and mother have different haplotypes, there is 1 in 4 chance that siblings will inherit the same maternal and paternal haplotypes and therefore be histocompatible with each other. None of the offspring will be histocompatible with the parents. As a result of recombination and other mechanisms for generating mutations, it is rare for any two unrelated individuals to have identical set of HLA genes. This is all about the haplotypes. And let us see the MHC molecules and their respective genes. MHC Class I and Class II MHC molecules are membrane-bound glycoproteins that are closely related in both structure and the function. Both Class I and Class II MHC molecules have been isolated and purified, and the three-dimensional structures of their extracellular domains have been determined by X-ray crystallography. Both types of membrane glycoproteins function as highly specialized antigen-presenting molecules that form unusually stable complexes with the peptide ligands, displaying them on the cell surface for recognition by T cells. In contrast, Class III MHC molecules are a group of unrelated proteins that do not share structural similarity and common function with Class I and Class II molecules. The Class III molecules include complement proteins. I am coming to the structure and peptide binding affinity functions of the Class I MHC molecules. Class I MHC molecules contain a 45 kilodalton alpha chain which is associated non-covalently with 12 kilodaltons beta 2 microglobulin chain. These are forming the Class I MHC molecules. The alpha chain is a transmembrane glycoprotein encoded by polymorphic genes within the A, B and C regions of the human HLA complex and within the K and D regions of the mouse H2 complex. See here we are using the alpha 1 and alpha 2, here the alpha 3. This is the beta microglobulin. Beta 2 microglobulin is a protein encoded by a highly conserved gene located on a different chromosome. Association of the alpha chain with the beta microglobulin is required for the expression of Class I MHC molecules on cell membranes. It is very compulsory. The alpha chain is anchored in the plasma membrane by the hydrophobic transmembrane segment and hydrophilic cytoplasmic tail. Structural analysis have revealed that the alpha chain of Class I MHC molecules is organized into 3 external domains, namely alpha 1, alpha 2 and alpha 3. each containing approximately 90 amino acids in length. A transmembrane domain is of about 25 amino acids in length, followed by a short stretch of charged, otherwise hydrophilic amino acids, and a cytoplasmic anchor segment of 30 amino acids in length. Beta 2 microglobulin is similar in size and organization to the alpha 3 domain. This is a beta 2 and this is alpha 3. It does not contain a transmembrane region and is non-covalently bound to the class 1 glycoprotein. So that is alpha chain, which has 45 kilodaltons molecular weight, alpha 1 90 amino acids, alpha 2 90 amino acids, alpha 3 90 amino acids, and there are 25 amino acids which are hydrophobic that form the transmembrane domain, and the hydrophilic one which form the cytoplasmic tail with the 30 amino acids, and beta 2 microglobulin which is looks like the which is similar to the alpha 3. It is also made up of the 90 amino acids and which weighs of about 12 kilodaltons molecular weight. Since data reveal homology between the alpha 3 domain, beta 2 microglobulin, and the constant region domains in immunoglobulins, hence alpha 3 and beta 2. Alpha 3 and beta 2 are members of the immunoglobulin superfamily. Immunoglobulin superfamily. The enzyme papain cleaves the alpha chain just 13 residues proximal to its transmembrane domain and releasing the extracellular portions of the molecule consisting of alpha 1, alpha 2, alpha 3 and as well as the beta 2 microglobulin. Purification and crystallization of the extracellular portion reveal two pairs of interacting domains, a membrane distal part made up of alpha 1 and alpha 2 domains, a membrane proximal pair composed of alpha 1, alpha 3 and the beta 2 microglobulin domains. The alpha 1 and alpha 2 domains interact to form a platform of 8 anti-parallel beta strands spanned by two long alpha helical regions. The structure form a deep groove. This is the deep groove. Are cleft approximately 25 angstroms into 10 angstroms into 11 angstroms with the long alpha helices as sides and the beta strands of the beta sheet as the bottom. This peptide binding cleft is located on the top surface of the class 1 MHC molecule and it is large enough to bind a peptide which is of about 8 to 10 amino acids in length. So this is the peptide red one. This peptide binding cleft is located on the top of the surface of the class 1 MHC molecules and it is large enough to bind a peptide which lengthens of about 8 to 10 amino acids. The alpha 3 domain appears to be highly conserved among class 1 MHC molecules and contain a sequence that interacts with the CD8 membrane molecules present on the cytotoxic T cells. Beta 2 microglobulin interacts extensively with the alpha 3 domain and also interacts with the amino acids of the alpha 1 and alpha 2 domains. The interaction of both beta 2 globulin and a peptide with a class 1 alpha chain is essential for the class 1 MHC molecule to reach its fully folded conformation. Assembly of class 1 MHC molecules is believed to occur by the initial interaction of beta 2 microglobulin with the folding class 1 alpha chain. This metastable empty dimer is then stabilized by the binding of an appropriate peptide to form the native trimeric class 1 structure consisting of the class 1 alpha chain and the beta 2 microglobulin and a peptide. This complete molecular complex is ultimately transported to the cell surface. In vitro experiments indicate that in the absence of beta 2 microglobulin, the class 1 MHC alpha chain is not expressed on the cell membrane. This is illustrated by Dandy tumor cells which are unable to synthesize beta 2 microglobulin. These tumor cells produce class 1 MHC alpha chains but do not express them on the membrane. However, if the Dandy cells are transfected with a functional gene encoding beta 2 microglobulin, class 1 MHC molecules appears on the membrane. Then coming to the class 2 MHC molecules. See here, class 2 MHC molecules contain two different polypeptides. Two different polypeptide chains. One is the alpha chain, which is of about 33 to 34 kilodaltons, and beta chain, which is of about 28 to 29 kilodaltons molecular weight, which associated by non-covalent interactions. Class 2 MHC molecules are membrane-bound glycoproteins that contain external domains, a transmembrane segment, and a cytoplasmic anchor segment. Cytoplasmic anchor segment. See here we are using, this is the alpha chain, beta chain, this is the transmembrane region, and this is the cytoplasmic anchor segment or tail region. Each chain in class 2 molecules contain two external domains, alpha 1 and alpha 2 in the alpha chain, and beta 1 and beta 2 in the beta chain. Membrane proximal alpha 2 beta 2 domains, membrane proximal alpha 2 and beta 2 domains bear sequence similarity to the immunoglobulin fold structure, so that the class 2 MHC molecules are also classified as the members of immunoglobulin superfamily. The membrane distal regions, that is alpha 1 and beta 1 domains, form the peptide binding groove, peptide binding cleft for processed antigen. This cleft is an open packet or open-ended groove due to lack of conserved residues. This cleft is large enough to bind a peptide which lengthens of about 13 to 18 amino acids. 13 to 18 amino acids. Several hundred different allelic variants of class 1 and class 2 MHC molecules have been identified in humans. An individual may express only 6 different class 1 molecules and up to 12 different class 2 molecules. Thus, peptide binding by class 1 and class 2 molecules does not exhibit the fine specificity characteristic of antigen binding by antibodies and T cell receptors. A given MHC molecule can bind numerous different peptides, and some peptides can bind to several different MHC molecules. Because of this broad specificity, the binding between a peptide and an MHC molecule is often referred to as promiscuous. Both types of MHC molecules, peptide ligands, are held in largely extended conformational that runs the length of the cleft. The peptide binding cleft in class 1 MHC molecules, the peptide binding cleft in class 1 MHC molecule is blocked at both ends, whereas the cleft is open in class 2 MHC molecules. Class 1 MHC molecules bind peptides that typically contain 8 to 10 amino acids residues, while the open groove of class 2 molecules that can accommodate of about slightly longer peptide which contains of about 13 to 18 amino acids. Another difference is that class 1 binding requires that the peptide contains specific amino acid residues near the amino and carboxyl termini. There is no such requirement for class 2 peptide binding. And coming to the MHC peptide interaction. Class 1 MHC molecules, see here there are certain statements. MHC molecules show broad spectrum for peptide binding in contrast to the fine specificity of antigen recognition by antibody. Peptide MHC interactions are non-covalent and mediated by residue both in peptides and in the clefts of MHC molecules. Each MHC molecule binds one peptide at a time but can bind many different peptides. MHC molecules do not discriminate between foreign peptides and the self peptides. Class I MHC molecules bind peptides and present them to CD8 T cells. CD8 T cells. In general, these peptides are derived from endogenous intracellular proteins that are digested in the cytosol. The peptides are then transported from the cytosol into the cisternae of the endoplasmic reticulum, where they interact with class I MHC molecules. This process is known as cytosolic or endogenous processing pathway. The bound peptide should be 8 to 10 amino acids in length, most commonly 9 non-americ peptide. This peptide length is most compatible with the close-ended peptide binding cleft in the class I MHC molecule. All peptides that bind to class I MHC molecules contain a carboxyl terminal anchor made of hydrophobic residues, that is especially leucine, isoleucine. Another anchor is often found at the second or third position at the amino terminal end. Whereas if you observe the class II MHC and peptide interactions, class II MHC molecules bind peptides and present these peptides to CD4 T cells. In general, these peptides are derived from exogenous proteins, either self or non-self, which are degraded within the endocytic processing pathway. Peptides derived from digestion of membrane-bound class I MHC molecule often are found to class II MHC molecules. Peptides recovered from class II MHC peptide complexes generally contain 13 to 18 amino acid residues, somewhat longer than the non-americ peptides. The peptide binding cleft in class II MHC molecule is open at both ends, allowing longer peptide to extend beyond the ends like a long hot dog in the bun. A central core of 13 amino acids determines the ability of a peptide to bind class II MHC molecules. Longer peptides may be accommodated within the class II cleft, but the binding characteristics are determined by the central 13 residues. The peptides that bind to a particular class II molecule often have internal conserved sequence motifs, but they lack conserved anchor residues. See here, the macrophage with the class II MHC molecules presents its antigen to the CD4 T cells, whereas a cancer cell with MHC Class I molecules present its antigen to the CD8 T cells. One is from the exogenous pathway, another from the endogenous pathway. These are the differences in between peptide interaction and the MHC molecules. And coming to the Class III MHC molecules, MHC Class III codes for genes unrelated to cell-mediated immunity, and these are codes for components such as tumor necrosis factor, HSP means heat shock protein, and complement proteins like complement 2, complement 4, and factor B. These all are the products of the Class III MHC genes. And this is the 3D structure of the tumor necrosis factor. And these are all about the heat shock proteins, HSPs. And this is all about the complement system and the complement proteins like the C1, C2, C3, C4, C5, C6 like that. So this is all about the MHC Class III molecules. And coming to the functions of the MHC molecules. Class I MHC molecules, they are the markers that display unique characteristics of self molecules and regulation of immune reactions required for T lymphocytes. Class II receptors that recognize and react with foreign antigens located primarily on macrophage and B cells involved in presenting antigen to the T cells. And Class III They are secreted complement components like the C2, C4, TNF and the heat shock proteins. And furthermore, some of the functions, MHC molecules are located with a bit of sample peptide fragment derived from the degradation of proteins present inside the cell. This peptide is a mirror image of proteins present inside the cell. MHC molecules contain self as well as non-self antigens, foreign antigens. They bring about defense against infections and diseases. They mediate certain autoimmune diseases. They are also responsible for individual smell of the people. And biological functions of the MHC include the differentiation and maturation of T cells to form the functional T cell repertoire, present antigen to initiate immune response with a phenomenon known as MHC restriction. That is, endogenous antigen is presented to CD8 T cell by the MHC Class I molecule. It is restricted. MHC Class I molecule should present its antigen to CD8 T cells only, cytotoxic T cells only, whereas the exogenous antigen presented to CD4 T cell, that is nothing but the helper T cell by the MHC Class II molecule. So there is a restriction of the MHC in presenting the antigen, processed antigen. So this is all about the MHC molecules and their gene complexes, type 1, type 2, type 3 molecules and the functions of the MHC molecules. Thank you one and all.